Hello everyone, and welcome to our second live link from Boulby Mine. Here we are, a kilometre underground, well into MINAR 5, Mine Analog Research. So what are we doing down here? Why are we a kilometre underground with a bunch of scientists from all over the world, scientists from America, Europe, and other places who have come here to test technology, do science and education for the exploration and settlement of space? Well, the reason why we're a kilometre underground is because when robots and explorers eventually go to the Moon and Mars, they may want to go underground into lava tubes and into caves. On Mars, some of these deep underground environments may be the best places to search for life, but they're also less extreme than the surface. So they may be good places to hide out from the extreme conditions on the surface. We're also underground here because we're in an active mine. This is a salt mine, a potash mine, where salts are being mined for fertilizers and other industrial uses. And so we're testing out instruments in a place that could be useful for mining. So planetary scientists, when they go to the Moon or Mars, they want to build small, rugged instruments that will work in extreme environments. And these are exactly the sorts of instruments that miners might like to use as well, for looking at ore, for trying to figure out where the best places are to mine. So by bringing planetary scientists testing instruments down here a kilometre underground, we can make a direct connection with a working mine. There's also fascinating science reasons to be here. We're actually in 250 million-year-old salts that were laid down in the Permian in a giant inland sea. And these salts contain within them interesting microbes living in brines, which is water that has seeped through the salt and created these salty solutions. So part of the reason for being down here is to understand life deep underground. A great percentage of life on Earth is actually underground. You and me are used to seeing our fellow human beings and cats and dogs, but that's only at our scale. Most of life on Earth are tiny microbes living deep underground. So we're here trying to understand the microbiology. So we've got science going on here, we've got technology going on here, and we've also got education. One of the things that we can do down here is develop excellent educational materials like curriculum lesson plans for school children across the world who may be interested in learning about science and learning about astrobiology and the search for life elsewhere. So we have educators here as well. We're using this amazing lab. We'll walk you through that in a moment. You'll see what's going on here. It's not just astrobiology. It's run by the Science and Technologies Facilities Council, who established this unique facility underground. So here we are in the world's first underground astrobiology facility doing this analog research. And just to explain what's an analog, you might think, mine analog, what's the analog? Analog means an environment that in some ways is similar to extraterrestrial environments that we might be interested in studying, such as, for example, the Moon or Mars. So this is an analog to the underground environments on Mars, and environments where there may be salt. Later on, we'll be talking to ESA astronaut Matthias Maurer, who is here training with us from the European Space Agency and learning about how you sample in extreme conditions. So if you have any questions for an astronaut, I'll be chatting with him later. Get those questions out, send them by comments, and we can answer some of your questions in about 40 minutes from now. So let's have a walk through to the first part of our astrobiology facility down here in Boulby Mine and have a look at what we call the Mars Yard, which is a place where we can test technologies for the exploration and settlement of the Moon or Mars. We're walking away from the main lab. We'll be returning to the lab a little bit later and now going into this quite dusty, salty environment that in some ways is similar to the dusty and some places salty environments on Mars. On the right-hand side, if Jen swings around there, you can see a little miniature porta cabin that we have. This is an extension to the main lab where we can operate rovers, test technologies. Let's carry on down here through into the Mars Yard. As you can see, we've got some busy scientists. And we're going to go and have a look first at a robotic rock hammer. So when you go out into the field, you want to break up your samples. So we have a look over here, you can see some samples. See, these are rocks that have been collected from different parts of the mine. We are looking at their geochemistry, their geology. You can see a piece of potash there. That's potassium chloride. We go over here, we can see some halite. That's simply sodium chloride. It's actually the same as your table salt, but it's full of organic material and different minerals. That's why it looks dark. And then over here, you can see some sulfate minerals. There are a lot of sulfate salts on the surface of Mars, and we also find sulfate salts here in Boulby Mine. And we're interested to know, what are the geological properties? Could there be signatures of life in these ancient quarter-of-a-billion-year-old salts? So we get these samples, and the first thing we want to do is break them up, right? We're on the surface of Mars. We want to break up the samples and get our sample to look for life. And we have an instrument that will allow you to break up a sample using a robot, and it's right over here. So let's have a wander over to the split instrument. It's Emma hard at work, one of the teams in Boulby, one of the members of the Boulby team. She's one of the people that keeps this whole thing running and operating. And here we are over at the split instrument. And what you can see here is this instrument breaking away at a rock as if it's on the surface of Mars. So I'm going to try and grab one of these guys if they have a couple of minutes. John, is this a good time to talk to you?
It is indeed, yes.
So we're going to talk to John Holt briefly from the University of Leicester, who is building this instrument. I'm going to give John a microphone there. So John, tell me what's going on here. What's the instrument that you've got?
Let me just pop this on here. Okay, so this is split. We call it the Small Planetary Linear Impulse Tool. And this is a device really for...
Let's hold it closer.
Sorry?
Let's hold it closer so we can hear you.
Hold it closer, okay. So this is the Small Planetary Linear Impulse Tool, a device for breaking rocks on the surface of Mars. And it works just like if you like, a jackhammer drill, the sort of thing that you might see somebody working with out on the road. But this is much, much smaller, not so much energy. And what we're trying to do is exploit the brittle fracture nature of the rocks so that we can then break them open. Now, I don't know whether Jen can get the camera down to here, but this was a section that we broke yesterday, and as you can see on the surface, the rock is really dusty and contaminated, and that's not good. But what you can see here is nice, clean, pristine material, and that's what the scientists really want to get to, so that we can look inside the rock. So this is being developed at the University of Leicester, funded by the UK Space Agency. And this is our third generation prototype. So everything that you can see here is similar to what we expect a flight device would look like on a future mission.
Great, thank you very much, John. So this is fantastic equipment that hopefully will one day go to Mars, and you can see the prototype operating here. And one of the advantages of being, you might say, why come here a kilometer underground? Why not just do this on the surface? Well, one of the major reasons for doing this is to create a team of people collaborating. So the samples that we just looked at, we break them up using split. We'll see some of the instruments they're analyzed by. So it's not just about the location, it's also about bringing people together in one place to do planetary analog research. And as I say, in the context of a working mine. So this is the first step. We've got a bit of sample here. Now let's go and look and see what we might do with that sample. We're going to wander over here to another instrument called the HABIT instrument, which is already approved for flight on the ExoMars Mars rover, the European Space Agency's rover. It's going to be flying to Mars. We can't really see it very clearly. The reason why is because we're down here in the extreme environment of Boulby, and Wi-Fi doesn't go everywhere, unbelievably. In fact, it's amazing we have Wi-Fi out here from the main lab. So I'm going to call over the team here, and they're just going to tell us something about the instrument. Yes. So Alvaro here from Luleå University of Technology. Alvaro, tell us what you're doing over there.
Yes, so there we have the HABIT instrument engineering module. It's the first version of the instrument, and we are testing here on the mine as a Mars analog. And we are basically testing different salts because, well, the main objective of HABIT is to characterize the habitability of the ExoMars 2020 surface platform landing site. And for that, we are going to measure several parameters, UV radiation. We are going to measure as well the wind speed, air temperature, ground temperature. But apart from that environmental part of the instrument that is quite important for habitability, we are also going to carry with Habit an experiment that is called Bottle. And the idea of this instrument is to measure how much water, liquid water, could be absorbed from the Martian atmosphere through several salts that are quite typical on Mars surface and that has a characteristic that they are highly hygroscopic. So for them, it's very easy to absorb water from the atmosphere, and we are going to measure that absorption with the instrument. And that is actually what we are testing here with several salts we have taken here in this salty mine.
So the idea is that down here, the environment around salts creates a particular humidity that can, in some sense, replicate the sort of environment you might have, a salty environment on Mars or a dry environment. So
exactly.
You're testing the limits of this instrument down
exactly.
here.
Limit, endurance as well, operations as well, because at the end of Mars you cannot fix, if you have some kind of error in your operations, you cannot fix it. Yep. So we want also to test those operations in a representative environment, yes.
And I should say they have a really excellent team. It's Ashwin and Miracle here. They've been working hard for the last week and a half. And they have more than just Habit. So as well as the instrument that's going to go on ExoMars, they've been testing particle detectors. So looking at the particles that come down into the subsurface here. They have an environmental sensor suite that's going to stay down here for many months. So as well as coming down here just for 2 weeks, we can use the Mars Yard to set up long-term experiments that can be run in this unique underground facility with internet connection, so they can check their instruments over many months rather than having to come down here. And they've also been doing three-dimensional mapping. So one day, if you go into a lava tube or a cave on Mars, right, we want to be able to map that environment. Do you want to say something briefly about your 3D mapper?
Yes, I think my colleague Tasmin can talk better about that.
Hi. So we are doing 3D mapping with RGB cameras and depth cameras, by which we use infrared beams to measure the distance between points, and we generate an image. So this method is an alternative to the LiDAR systems that we have in the market. LiDAR systems are quite expensive, and this is a low-cost approach for mapping. And since we're using infrared, this can be done at night. Yeah. You don't need any lights. So this is one of the best methods for mapping. So,
Good, thank you very much. And yes, last week they were down here with their 3D mapper, and it was amazing. They came in here in complete darkness and mapped this area, and you could actually see the rocks appearing out of the darkness of the computer screen as their infrared mapping worked over there. And you can imagine going into a lava tube or a cave on Mars. It's completely dark. You may not be able to light the whole thing, or at least that might be difficult to do. So if you can have an instrument that will allow you to instantly map in 3 dimensions the area ahead of you, you can start planning your EVA, your extravehicular activity into this cave environment. So some very exciting technology being developed here by the group at LULO, and we hope that these instruments will be down here monitoring this environment for a long time to come. So thank you very much, guys. See you later. We're going to move back towards the main laboratory now and start to see what's going on in the main lab, and more than just the astrobiology, some of the other science that's going on down here in Boulby. So you can see that the Mars Yard is very versatile. In future iterations of MINAR, mine analog research, we plan to bring rovers down here and to operate them from the surface. One of the exciting things that happened last week is we conducted a very simple test of an EVA, an expedition team heading out onto the surface of Mars from the surface, communication from the surface to the subsurface through the internet link into the mine to test out some of the operational constraints of people working underground in lava tubes or caves on the surface of Mars. So we can use the Mars Yard to test these various instruments and rovers. And as I said earlier, we're also interested in the science. Of course, that's what drives all of this. What sort of life can live underground? And so by coming into our Mars Yard, we can test out environmental sensors. In fact, the environmental sensors that the LULO group are developing are going to be buried in an experiment where we're looking for microbiology and life underground. So we'll put those sensors under the salt and we can see what sort of conditions the microbes are exposed to. So as well as the technology for future planetary missions, it's also used to do scientific investigations of life down a kilometer in these salts, subsurface microbiology. So all of these things are connected. We're going to wander into the main lab now. As I say, this was developed by the Science and Technology Facilities Council. It's one of the few underground labs in the world. There are only a handful of these facilities underground where you can do science over a long time period. This is one of them. It's been running now for over a decade and allows us to carry out scientific investigations of astrobiology and dark matter. And we're going to go through and see what's going on through here. There's Lou over there. I'm going to embarrass her. She's the one that makes all of this happen. We just turn up as scientists. She's the one that does all the logistics, gets everyone down here, and it's a huge effort to bring 20 or 30 people underground a kilometer and to make all this work smoothly. So thanks to Lou. Moving through here. And here we see some educators hard at work. And like I mentioned earlier, education is a crucial part of this. We're doing really interesting science and technology testing that allows us to develop classroom activities for kids to learn about deep subsurface science and some of the exciting things going on. So I'm going to call Audra over here, who's leading the education team. Audra, what are you doing this morning? Tell us what you're up to.
Hi, Charles. So I'm here with a group of teachers. We're from all over the world. So I'm a U.S. citizen, but I work in an international school in Azerbaijan. We have Lalit, who works in an international school in Armenia. We have Libby, who works in an international school in Belarus. And we have Anna Lee, who is a writing instructor in Australia. And so what we're doing is we're working hands-on with the researchers. We're learning about their field methods. We're learning about their field techniques. We're learning about the importance of using this area as a Mars analog, both content and methods. And then we're taking all of that information that we're learning and we're trying to break it down into some simple lessons and activities that we can give teachers as resources. And so one of the things we're focusing on is using an EVA, an extravehicular activity, that astronauts would use if they're on the moon or on Mars to explore their area and to gather samples. And so we're trying to come up with an activity that students can use, that students can do to be excited about science and to be doing these field methods that researchers are doing.
So the idea is that you can sit in a classroom or you can sit in an office and write a lesson plan, but the real value here is being able to be embedded within a group of scientists and learning about what they're doing.
Yes.
And develop lesson plans that really reflect real science going on and technology testing.
Yes.
Would you say that's one of the main?
Yeah, absolutely. So as we're learning from the researchers current methods and current research, we can take that to students so students can be doing actual authentic science.
So the expedition you had earlier on in the week where some scientists just went out and practiced sample collection, the idea is you'll turn this into some sort of lesson plan where children in classrooms can pretend to be on an expedition on the moon and Mars and split up into groups and think about what they're going to go and do out there in the field.
Yes, exactly. So we were doing a run through so that we could figure out all the steps of it, and we want students to actually be doing these simulations of Mars exploration trip.
Yeah, and it's sort of essential, right? Because otherwise we're down here doing science and testing technology, which is great for us, but it doesn't really help the younger generation. But if we can do education here, then we have a way to use this to educate school children about space sciences and technology and other things. And so there's a sort of legacy of building the next generation of space scientists. Some of you will hopefully one day go to the moon or Mars. So we just come a kilometer underground and hope that one day we'll go to the moon and Mars, but probably unlikely, apart from Matthias over here, who is a European Space Agency astronaut, and we'll be talking more to you a bit later. But we'll have a chat a bit more detail. But what do you think about training the next generation of astronauts and space scientists with this sort of activity?
I think we absolutely need more explorers to venture out into space and to discover all the secrets that are out there. And so we need to start early training them, and I think your approach is highly valuable for us. Thank you. I wish I would have had your training early on as a kid.
Yeah, I sort of agree with that. Actually, I was saying this morning to someone that we never really had these possibilities of doing astrobiology. Astrobiology didn't really exist when I was in school. It was a recent thing that happened in the 1990s. So having the possibility of now taking what we've learned and turning it into lesson plans seems like a really valuable thing to do.
Yeah.
Great. Well, thanks a lot, Audra, and thanks, Matthias. And we'll be talking to Matthias more a bit later about his training here and what he's learning from all of us about sample collection and deep sorts. Great. Great. Let's carry on through to the main lab, where we're going to see a little bit more of the astrobiology and the instruments, but we're also going to talk about dark matter and some of the other things that go on deep underground. So if you come through here, one of the crucial things about a dark matter lab is it's got to be really clean, because we can't have dust. I'm now going to I'm going to try and do this as smoothly as possible. Dust and general particles inside the main lab. So we wear one of these suits. It's a bit of an exercise, this. It's one of the exciting things about working in an underground lab. Preparing is relatively new. We approached the lab here. They said, "What about underground lab?" They were enthusiastic. So we got got a kilometer of rock above us, and that means that radiation is blocked out, and it makes it easier to look for particles of high energy particles interacting from the dark matter, interacting with the normal matter that you and I are familiar with. Yeah. You can see this is a bit of an exercise. And here we see, and down here at all times. It's strange thing. Scientists walking around clean labs with hard and just shows you an interesting interface between miners and scientists. Strange meeting minds of groups of. This is a mine given potash. Overseas, Israeli Chemicals Limited. I think, why would a mine have been a really inspirational company in have a little look at a Raman instrument. So over here we have the Raman instrument. So we've got our samples, as you saw outside. We've split them up, and now we've got some bits of rock that may have been collected by a rover or an astronaut. We want to do something with them. We want to analyze them. And one instrument that allows you to analyze samples is called a Raman spectrometer. So if I let you come in the cupboard here, we're going to meet Anushree and David here from the NASA Ames Research Center, who are operating the Raman instrument. So guys, you want to tell us what's going on here?
Okay, hi guys, this is Anushree and this is David. I'm working with David Wilson. So this is Raman laser spectrometer, and we're testing some of our very important ancient samples of different like polyhalite, halite, potash. So yeah, we're getting good peaks, and that's how you identify your sample, the composition of your sample, and which is very important to also identify and characterize the biosignatures trapped in those samples. So that's why it's very important.
And you can see on the screen there a couple of peaks.
Yeah.
So all the minerals have different different peaks depending upon what they are. And I think you have the laser is off, but when it's on, you fire this at your sample. And what it does is it sends light out, the light comes off right, it scatters off, and it's almost like a fingerprint. If you think about it like forensic science, it's like a fingerprint of your particular mineral.
Yeah, of course.
So that's really exciting. And this is a fairly big instrument. We're not trying to carry it around, but there is a lot of research going on into building handheld instruments now, small ones you can put on a rover or take with you as a human explorer.
So as you suggested that this is going to be first time you going to be used here in this mine. Mm-hmm. So it's very important, yes, the research.
Yeah, good. Thanks a lot, Annie. I'll take the
yes.
microphone off you and we'll move through the main lab. So that's just one instrument you can see that can be used to study samples. So we're going to move through here. So now you can see we're transitioning from the main lab area into the main lab area from the less clean area. As we go through here, we're going to go and have a look at matter research that's going on next door. So this is the main lab. You can see it's a really impressive facility, very large lab. It goes right the way down to the end there. We have a whole range of experiments, and we're going to go and have a look at just a few of these, just briefly. So, Chris, do you want to come and tell us about the drift experiment? What's going on here?
So as you've been saying, we have been looking for dark matter now for well over a decade here in Boulby. And as you said, Boulby is an ideal place to look for dark matter because if dark matter exists, and it might not, but if it does exist, it will be a very, very quiet whisper in what is a vast noise, shouting noise of radioactive noise. So Boulby, we call it a quiet place in the universe, radioactively speaking, because only down here can we listen in for these very faint whispers of dark matter signals.
Okay. So this is basically the detector.
That's right. So what you can see behind me, these two steel vessels are the detectors themselves. This is called drift. Drift is a very special detector in that it's a directional detector. So I call this a very clever detector. Most detectors are like magic boxes, and they look for ping signals. So we are looking for dark matter particles. So these are very, very small subatomic particles that we think make up a huge quantity of the mass of the entire universe. So these are crucial particles that we know very little, even nothing about. So we hope that these small particles will eventually interact with matter by simply knocking into the nucleus of an atom. So this very, very unlikely and very small cross-section chance will eventually happen, and we hope that it happens inside one of these detectors. In a normal detector, when this happens, you get a ping response, so just like a counting mechanism almost, that you can see them. Drift is a little bit different, and instead of just getting a signature that you've seen something, drift tries to get a bit more information. So instead of just having a point, you'll also have a track, and from that you can get a direction, and that's why it's called a directional dark matter detector. So that's why it's a bit special.
Great. And Chris, the reason for being down here is to use that kilometer of rock above us to essentially screen out that background noise of radiation from the sun and from the galaxy and from the cosmos generally. It's, as you say, a quiet place in the universe where the radiation's, that's the basic idea here.
That's right. To give you an idea, so a huge factor to this background noise is cosmic rays. So these are really high energy particles coming from exploding stars, so supernova, distant galaxies, and they're still firing their cosmic rays towards us. And for every million cosmic rays that the surface sees, we only see one of them. Okay. So it's a huge reduction, and it's a very cheap reduction by just coming down the mine. The lead equivalent would be huge amounts of lead. It would be costly and impractical. Coming down this mine is great for logistics, and we've got the infrastructure already, so it's a win-win for both the mine and for the science as a whole.
And obviously a major part of science is collaboration. Some people say, why do you come down here to do analog research? But even if there were no reasons to come down here and you could do all this Mars analog research on the surface, down here, astrobiologists are rubbing shoulders with astrophysicists thinking about dark matter, and there are other experiments besides. There's a new experiment that's going to start looking at how salt behaves geologically and whether we could store carbon dioxide down there as part of carbon dioxide capture and storage, carbon capture and storage. And so one of the benefits of this environment is collaboration between different groups of scientists and new ideas. Technology that we develop for geology or dark matter can have links to technology and astrobiology. So it's about a scientific collaboration that's going on here, as well as the technology itself. Thanks a lot, Chris.
You're welcome.
I'm going to move on to some more ExoMars instruments. So you can see dark matter has been the major legacy of this lab, and one of its major focuses over the last decade or so. So we're approaching a group here who are testing an instrument that has also been approved for the Mars ExoMars rover, the European Space Agency's rover, and it's called the CLUPI, the Close-Up Imager. If we come over here, we will see what they're up to. Hello, Jean-Luc.
Good morning.
So, Jean-Luc, this all looks very impressive, lots of interesting samples. Tell us what you're doing here.
Yes, so here we are testing a sample, and we are using a CLUPI for the close-up imager. This is the instrument. So we have a view here. It's like this. So actually, it's, I'm leading a very strong team, international team. And so this is a small camera. This is a very high-resolution camera in color. We are able to take your sweep. So you have model. This is model. And with this, you can take images from a walking distance from about 10 centimeters to infinite. So this is really high resolution color images. And this is really a link, part of all the investigation of all the instruments on board the rover.
Thank you, Jean-Luc. Thank you, Tommaso, as well. So here again is another instrument that's on a rover or with potentially a human explorer. You get your sample, you've analyzed it with Raman. You also want to get close-up images that are very, very high quality. And the CLUPI instrument, you can see here, a nice image here on the screen of a piece of halite from this mine with a calibration target. Calibration target is there so you can make sure that your photograph has the correct color. If the colors change slightly, then you can use the calibration target to correct and get it exactly as it would appear in the natural environment. You can see a very crisp, sharp image. We want exactly that sort of image from Mars, right? That's the idea. Hopefully on the ExoMars rover and future missions. I should say Jean-Luc here is being very modest. He's already had a camera that's been on the Rosetta mission, so he's already taken images from the surface of comets and now hopefully onto Mars. So let's carry on down here. Thanks, guys. And we're going to now go and talk to Jen Blank from the NASA Ames Research Center, who was working with a group that have now left, but were here last week, sequencing DNA. So DNA, the information storage system of life, we can extract this from microbes and from life underground, and we can sequence it. What does that mean? It just means looking at the letters of the genetic code, essentially those letters and writing them down and getting a sequence of those codes. And that tells you what sort of organisms that you have from your DNA. And last week, they brought down a miniature sequencer that managed to sequence DNA from this environment. It was the deepest DNA sequencing, in situ sequencing, that has been done to date, a kilometer underground. So I'm just going to go over here, and we're going to have a quick word with Jen. She's going to tell us about what she was up to last week, sequencing DNA. Jen, do you want to just tell us briefly about this MinION that you were using?
Actually, I'm working on something else right now, but I just want to show you, these are my our colleagues from JPL, and they came out, and they are the microbiologists, but they let me tag along with them, and we walked around places near the underground laboratory, both in and outside, and took samples, and then we used this really cool new sequencing device that once you prepare a sample, so we collected samples and then we extracted DNA from the soils, primarily the soils in traffic areas and quiet areas around the tunnels of the lab, looking to compare the microbial activity in populations in areas of high traffic humans and also in areas that might have no contamination by people. Anyway, and here's the device. You want to see it? So,
yeah.
It's actually been packed away right now. But it's called a flow cell. And did you describe it, Charlie?
No, I didn't. So
okay.
do you want to tell us how this works and what it is?
Well, I don't really want to contaminate it, but it's totally cool. This is the device, and inside you actually have a little flow cell to which you can extract your, or rather inject your DNA that's been prepared through a 50-step recipe In fact, our visiting astronaut Matthias has performed this activity in a training exercise already. And then we essentially just pipette our DNA from all the samples that have special magnetic tags on them, and they go through this flow cell, and we did sequence counting for 2 days, and the process finished early this week. But we have sequenced not microbes from the deepest part of the world, but this is the deepest in the earth that this actual lab activity has been performed. So it's pretty exciting that you can actually do analyses in situ, right? Yeah. So that's really exciting, Charlie.
Yeah.
So. we got great results. So it's good.
Yeah, I mean it's fun to make these new records. Of course, they're not. I should say they're not necessarily scientifically significant, but breaking those sorts of records is good for technology demonstration. It shows that you can push the limits of technology in new environments. I should say Jen is from NASA Ames, and she's been doing other work down here as well. She's interested in camera systems, looking at different types of minerals and how that might help us in the search for life on Mars. Thanks a lot, Jen. So we're going to head back now through the main lab. We're going to go back to the other side and have a little bit of a chat with Matthias Maurer, ESA astronaut, and find out what he's doing down here this week and why he's with us in Boulby Mine. So if you have any questions for an ESA astronaut, get on your computers now and start sending them across to us on the comments section of this live feed, and we can answer some of those questions. So you can see one of the advantages of being in Boulby Mine is we've got the side area, which is a sort of dirty area of the Mars yard, and then we've got this incredibly clean lab, class 10,000, that allows you to operate very sensitive, sophisticated planetary instruments that maybe are at an early stage, not ready to go out into the field, but you want to test them in a clean area and then work towards making them ready to go into more rugged, extreme environments. So both of those things are very helpful for astrobiologists. And we will be continuing this work for the rest of the week. During the last 2 weeks, we've had a few teams that have been here, coming and going. Not everyone has been here for 2 weeks, but we have people working on camera systems, the DNA extraction systems we saw before, and also developing new instruments such as fluorescent spectroscopy. So another person here is from St. Andrews University, and he's been looking at UV spectroscopy to try and understand how we can look for minerals and signatures of organics or life in rocks on Mars. So if we come back through here, we're now going to move out of the main lab area into section just beyond here, where we'll have a discussion with Matthias. So as you can see, we have to go through the opposite procedure here of removing the clean room suit. So the lab is an impressive facility because it allows us to bring people from all over the world to do this astrobiology training. So this week and last week we've had scientists from Europe, from the US, and also a connection with the Callum Center. The Callum Center was set up a few years ago in honor of Abdul Kalam, who is really the father of the Indian space program. Let me get this off here. And he was the person who really drove rocket technology in India and got the Indian space program up and running. And in honor of that, the Callum Center is trying to develop educational activities for kids across India to get them involved in space exploration. And they have been connecting with us through these live feeds. We've been interacting with many tens of thousands of Indian school kids to try and get them interested in our analog research. I think that was the most interesting experience I've had in Boulby Mine. But there we are, we've got there finally. And so let's move through into the section next door and have a chat with Matthias. Right. So, go to the thing. Oh, Matthias, do you want to come and join us? So we're going to go to the end of this lab and have a chat about different rock samples with Matthias Maurer. So if you just follow me to the end here. And soon he's going to join us, and we're going to have a discussion about some of the things that he's been doing. So over the last week, Matthias has been here as part of Pangaea, which is a European Space Agency astronaut training program. Hi, Matthias. Thanks for joining us. So do you want to tell me what you've been doing here and how you found the experience?
Well, it's for me the first time here in a cave. I'm already saying, like, I've been in caves for training, but actually it's the first time in a mine. And so far I'm following different scientists and following what they are doing down here, how they run their experiments, and I try to learn like different techniques and technologies that I hopefully one day will employ, once I'm on a foreign body.
So the idea is being part of scientists and going out into the field and collecting samples will help you understand what sort of samples you might collect.
Exactly.
What have you found? Have you found any?
Well, so far I found lots of salt, like you have shown here already, different types of salt that I've seen out in the field. And I mean, you're way more expert. You can summarize it. I believe that is the salt, the table salt.
Yeah, this is a bit of halite. You can see it's. I was showing the viewers earlier some of this material outside and saying, you know, it's table salt, but it would be white normally if it was on your table. But it's full of organic material and minerals, so it's got this sort of brown-black coloration.
So that I could spread on my salad.
Yeah, so you could eat that,
but I wouldn't have that one on my salad.
That's potash, it's potassium chloride, and that's the polyhalite you saw earlier. So what's the idea? You come here and you learn how to collect samples and which ones might be useful for scientists.
Yes. So the question that we as astronauts have, we have an EVA, an extravehicular activity. So we walk on the surface, and we are limited in resources. Let's say we have for 6 hours enough air to breathe, and in these 6 hours we want to do as much science as possible. So you want me to be quick in the field and say, like, okay, that's a good sample, I take it, my scientists will be happy, or I take that one, and you say, like, oh no, that's not the correct one. So that's the type of decision-making process that I want to learn here. Which are the good samples?
Okay.
And I understand you want to find life.
I do, yes.
So
that's really what I want to find.
Explain me again in a few words, which of these samples you want me to bring back to find life?
Yeah, so let's have a very brief chat about that, because there is something that's really important for you as an astronaut to know, and that's something called water activity. Have you heard of water activity?
Yeah, I've heard of water, but being active.
Water activity. So water activity sounds very dynamic. Actually, it's a technical term, and what it means is, in very simple terms, the availability of water. So, for example, if I've got this pure water here, almost pure water, we drink a lot of this down in the mine to keep hydrated. This has a water activity of 1. And something that's really dry has a water activity of 0. So it's a scale from 0 to 1. The higher you go, the more water is available.
So this is very dry to me.
Looks very dry, but if you mix it with water, this is actually a brine, and it looks clear. That's only because the salt's dissolved. This has a water activity of 0.75, which is about acceptable for some types of life. Some types of life can grow in this brine. If you push that water activity down, then life can no longer live in those solutions. So household honey, for example, has a water activity of about 0.55. It's a little bit below the lowest limits of life, and that's why you can leave honey on your kitchen table and it will not go moldy, because the water activity is too low for life. So some of these salts, if you dissolve them, will have high water activity, some of them will have low. So what you need to do as an astronaut is to pick the salt samples that when they dissolve will have a high water activity and could support life. And samples that have very low water activity when they dissolved in water may not be so good places for life. We'd still like those samples, but maybe not. So
let me see if I fully understood. So if I take this water here and I take a little bit of salt independent of that one, is there already a difference in water activity?
Yes, there will be. That's right. So when you dissolve the salt in the water, it will dissipate, and then the water activity will drop. So as you add more salt, water activity will drop down.
It's a question of the concentration of the salt and the type of salt.
Yeah.
All right.
I don't know whether we have any questions from viewers. One question's come in. They're asking, if you go to Mars, how much training will you need in geology and astrobiology to do your job there?
Well, you need to be trained, obviously, to be efficient. I need to understand what you want me to do, and there is not one single type of training that fits all purposes. Mm-hmm. So what we are doing, we have geological training. We go out into the field. That's a training that we run in Italy for sedimentation, and we also go to Lanzarote for volcanic training. And yes, that here in a mine is a special training because it's the first time that I'm involved in seeing where life can actually grow in these types of rocks and brines.
Mm-hmm. Okay, so it's very much about getting training in different parts of the world to look at these different sorts of environments. Another question I think has come in asking, why wouldn't you just go and train in many different environments around the world? Why particularly Boulby? What's the use here?
Well, I think training in many different places of the world is actually a good approach, but then the time of astronauts is also limited, and I cannot only train in as many sites as possible, but I want to go to the best places that are of interest for you guys. Like when going to Mars, you know already more or less like which sites could be of interest to find life. Okay. And I believe Boulby is one of these sites.
Mm-hmm. So I think it's true to say, right, would you agree with this, that there's no such perfect analog site? Some people ask, okay, you're in Boulby Mine and you're in all these salts, but Mars is volcanic. It's made of volcanic rock. It's not exactly like Mars. And I think what you're saying is, if I'm right, that many analog sites have different aspects that are useful for training an astronaut and for learning things, but nothing is perfect. So down here, we're covered in salt, thick layers of permeous salt, but of course Mars isn't just salt. But here we can learn about these evaporitic environments on Mars, whereas in a volcanic environment, like Lanzarote where you've been, then that's more of an analog to the lava tubes and caves that you might find in volcanic rock. So all of these things have something slightly different to offer.
And we also need to understand that on Mars we have a lot of radiation.
Yeah.
So a good place to look for life is underground.
Yes, that's true. So I suppose it's more protected, right, underground. Yes. So many of the things we're looking at in the salts might tell us something about whether there could be life underground on Mars. So hopefully when you explore Mars, you may go into lava tubes and look for salt deposits, perhaps.
Yeah.
Something to mention, which I think we talked about earlier, and for the viewers, some people might still be thinking, really, what's the link with Mars? But we know that there are salts on Mars. If you look in some craters on Mars, you find evaporite, essentially evaporated salt minerals. Even table salt like that is on Mars, halites. So if you went to some of these craters, you might find material very similar to this. And people think that these salt deposits were formed when you had bodies of water on Mars that evaporated. The water evaporated away, and the salt was left behind. So these could have been the last remnants of habitable conditions on Mars. So would it be true to say that as an astronaut, maybe you might go to these craters and look for salt look for these salt deposits and bring them back to scientists?
Yeah, that's actually a very good idea that we go to this area, and I believe Curiosity rover gave us this information. So thanks to this rover missions, we know where we would like to send rovers to. You had a question over there.
Question coming in, just a little bit of logistics here. Jen's going to read off the question.
Got a question from Sacred Heart Secondary School. Hi guys. So the first question is, how long would it take to get to Mars?
That's a question I believe I can answer. It depends a little bit the way we take. So the fastest way would be a mission of around 500 days out and coming back, so quite a long way, and it would only leave us for around 1 month staying on Mars. But there are also other ways that can bring us to Mars, but these ways are slower, so it can take up to 1,000 days for one mission. So a very long trip to Mars.
Thanks, Matthias. We've got one more question, probably for the both of you. Do you think life exists on Mars?
So Matthias, who's gonna stick their head above the parapet first? Do you want to say what you think?
Well, it's like I have not been to Mars, and we haven't found life on Mars yet. But I believe, like the universe system or the solar system has so many different places that are favorable to life. So I would be very, very surprised if we would not find traces of life. but that's just my gut feeling. And you're the specialist here.
No, I think you're right. I think it's more than a gut feeling. I think that we now know that there are environments on Mars that had liquid water. We know that water is one of the requirements for life. And we know that this water has been in contact with volcanic rocks. And you know from your work in Lanzarote that where you have water in contact with volcanic rocks, all those elements and nutrients come out, and they're usually very good places for life. So what we do know, which is a stunning difference from 20 or 30 years ago, is that there are environments on Mars that were potentially habitable to life, could have supported life. And the Curiosity rover has also shown that to be the case.
Yeah.
So I guess the next big question is, okay, they were habitable, but did they contain life? And that's still the big question in astrobiology. Are we alone in the universe? Is there life elsewhere? But I agree with you. I think we're at least closer to finding environments where we should look for life. And then even if we didn't find life, that would be pretty significant, right? If we found,
I believe a lot of people believe life is something like you or me, but I don't think I would like to live in that brine. So what kind of life do you expect in this liquid here?
Yeah, so mainly microbial life. And it's a very good question that the person asked, because what sort of life are we talking about? Exactly as you said.
It's not green in there, no?
It's not green in there. And in fact, if you look at that, you compare these two things, got your water and your brine, which one contains biology? Well, it's actually a bit of a trick question because they both contain biology. But the point is that these brines, they don't look like they contain lots of life. It just looks like water. But in fact, it does contain life. It contains microbes living deep underground that are using organic sources. So from your astronaut perspective, that's also another crucial thing, because it's not as if you can just see life. You know, we walk around the surface of the Earth, we see large animals. It's easier for most of us to be able to identify life. When you're an astronaut, probably the most likely type of life you'll find, if anything at all, is microbial life. And that's why we've seen some of these instruments as we've walked through the lab today. You need quite sophisticated methods to find that life. So I think that one of the benefits, of course, of linking all of these instruments with astronaut training is hopefully, you see how samples come through the sample chain in different rovers and possibly in human missions. I mean, do you think that's of value for astronauts to see these instruments actually operating together?
Definitely. It's always a value to talk to the scientists who run the experiments and to understand what's the purpose of the experiment, so that later on, when I'm their technician on the remote side, that I do the best to get them the samples that they need.
Yeah.
And I'm also looking forward this afternoon. We will have some experiments running UV light, which is usually used in CSI crime scene investigation type.
Yeah.
And I learned this morning that this is also a nice technique to find life maybe on a different planet.
Yes. Great. So there'll be extra training there. And I think this is part of a small part of your training, right? With Pangaea and caves at ESA, you're doing a lot more. This is just one type of analog environment, but hopefully adds to that sort of jigsaw puzzle of training, so that eventually when we rely on you so we just sit here and we hope to get samples back from other planets, you have the privileged position of maybe going and collecting these samples from the Mars or Moon for all of us to look at. So this is very much a collaborative effort as we push into space and hopefully eventually establish human exploration on the Moon and Mars. So I'd like to thank you, everyone, for taking part in this live link and joining us and Matthias to talk about some of the work that we're doing here as part of our Mine Analog program. We'll be back at 3 o'clock with another live link and maybe looking at some of your training on the fluorescence, I think, outside. So if you want to see Matthias doing some training and looking at samples using fluorescence methods, we'll try and organize that for 3 o'clock. Otherwise, thanks again for joining us here in the STFC lab, a kilometer underground, and we'll speak to you soon.
And send us questions.
Yes, send us questions.